use of rDNA technology in the field of agriculture. Now we'll start preparation of this biotechnological use in the field of medicine, then the diagnosis of different types of disorders, disease, etc. Now, one example of this rDNA technology is preparation of humulene. Actually, for any treatment of any disease, effective treatment, the diagnosis must be at the earliest stage. And for that, that is known as molecular diagnosis. So, for effective treatment, because we know the early diagnosis is required, so there are some techniques in rDNA technology which help in this process, like PCR, polymerase chain reaction, then ELISA test, that is enzyme linked immunosorbent assay, and these tests can diagnose the disease or the disorder at the earlier stage, like this ELISA based upon the antigen antibody interaction. When the symptom arise, till that time the concentration of this pathogen becomes high in the blood. So before that, that must be diagnosed, and there are these the methods, that is PCR, ELISA, etc. They will help in this process. Now, this antigen antibody interaction is the method for this ELISA, in which due to the presence of antigen, that is protein, glycoprotein, etc., or by the detection of the antibodies which are synthesized against that pathogen entering in the body. These are some methods for the detection of this disease. ELISA can be applied in different types of disease like cancer, AIDS, etc., and other diseases also. PCR also in the same way. So in this way, this biotechnology give us PCR or ELISA, different types of tests, different types of methods to test the disease, to diagnose the disease at the earliest stage. Now, one of the example of this biotechnology is preparation of humulene. As we know, insulin is a hormone which is required for sugar metabolism in the body, carbohydrate metabolism in the body. When the insulin is not in a proper way, it is not synthesized, then the person is said to be diabetic, or the person is suffering from the diabetes disease, and the sugar metabolism is not proper. Such persons, such patients require the supplementary insulin from outside. Earlier, this insulin was prepared or isolated from the cattle or pig's body, and that was allergic in nature, and that was not suited for all the patients. Then, because we are knowing the structure of the insulin, then nowadays, due to the help of this rDNA technology, we can make the insulin which is just by the help of the human cell, human body, and it is best suited for human body, and that's why it is known as humulene. Now, this insulin consists of two chains, that is polypeptide chain A and B, which are linked by the disulfide bridge. In human body, it synthesized as a prohormone, and it is in inactive form, but when it becomes mature, then it becomes active. So in prohormone condition, it along with this chain A and chain B, it also having C peptide bond, but it will be removed. It is not present in the mature insulin. By knowing the structure of this insulin, one Eli Lilly company that is an American company, in 1983 was able to synthesize the DNA sequence of polypeptide chain A and chain B of insulin. Then these two chains were introduced in the body of the plasmid of E. coli bacteria to produce the insulin. Now, after making this chain A and B, they were combined by the disulfide bond, and that was resulted in formation of the insulin known as humulene, which is best suited for human body. So we can see it is very helpful for treating the diabetic patients. Now, next example is gene therapy. Gene therapy, actually this is a technique which is helped by, or this is the technique belongs to biotechnology, and this allow the correction of defected genes. When there is any defect of gene, due to that some disorders will be there, so that can be checked, that can be corrected, and this technique is known as gene therapy. One of the disease that is known as ADA deficiency. It's the disorder due to the gene, genetic disorder, and in this ADA, that is adenosine deaminase, is required for the proper functioning of our immune system. When this gene is lacking, then this can be checked, or this was first corrected in 1990 in a 4-year girl child, and this was the first gene therapy. For treating this disorder, there are different methods were there. First was the bone marrow transplantation, then enzyme replacement therapy. That means ADA enzyme is in the form of injection. It is introduced into the patient's body, but these are not permanent cure. Then gene therapy. Nowadays, with the help of rDNA technology, we can apply this technique, that is gene therapy. In this gene therapy, the lymphocyte from the patient blood is taken out, isolated, and cultured outside the body in the laboratory, where a functional ADA gene is introduced by the help of the vector, that is retroviral vector. After introducing that, lymphocyte is again returned to the patient's body, and then this method requires periodic infusion of this lymphocyte. This is not also permanent cure. This requires periodic infusion. But if the gene can be isolated from the cell at the early embryonic stage, and at that time if ADA is transplanted or introduced into the body in early embryonic stage, then it can be the permanent cure of that disease. So in this way we can see there are different helpful things, there are different use of biotechnology in the field of therapeutics. That means preparation of different types of therapeutic drugs, then diagnosis of disease at earlier stage so that effective treatment can be given. At global level, about 30 effective therapeutic drugs are there which are in use, and in our country, in India, it is about 12 which is now in the market, and that is the result of this biotechnology, rDNA technology.
This is showing the use of biotechnology in production of humulin. This is the structure of proinsulin, that means chain A, chain B, and this is the C peptide, which is in mature form. It is removed and only chain A and B is present, and that is made by the help of biotechnology and by the use of the bacteria E. coli, that is known as humulin. This insulin or humulin is not allergic for the person who are suffering from diabetes. Then this diagram is showing the introduction of this desired gene in this plant. For that, this is the source is Agrobacterium tumefaciens, and from here bacteria is gene is taken, foreign gene. This is the recombinant plasmid. It will enter into the bacteria to make multiple copies, and then this bacteria is used to insert the tDNA into the plant cell grown in the culture, and this gave rise to this plant which is generated from a cell clone. So these are the application of biotechnology. This slide is showing the gene therapy in the field of pharmaceutical. Gene therapy gives the patient a healthy version of a defective gene. If any person has, any child has defect in their gene, then that defected gene can be corrected by this therapy, gene therapy. Here we can see the cell has defective gene, healthy gene is introduced here, and cell functions is restored. The cells are removed from the patient and then it is cultured in the laboratory. A virus is altered so that it cannot reproduce. A gene is inserted into the virus, and this altered virus is mixed with the cells from the patient. The cell from the patient become genetically altered, will be these are injected into the patient body, and the genetically altered cells produce the desired protein or hormone which was earlier lacking. So these are the steps of gene therapy. This is the comparison between conventional therapy and cell and gene therapy. One of the example we have discussed about ADA deficiency, which was cured by this in 1990 by this gene therapy. In conventional therapy, uses small molecules, peptides, proteins, etc., whereas gene therapy uses DNA, RNA, cells. Then this is chronic therapy, this is one time treatment. That means here again and again injection or infusion or pills is required, whereas here this is effect, the treatment may be permanent after a single administration. That's why it is known as one time treatment. Now manage or treat symptoms long term, here potentially curative, and in this way these are the comparison between conventional therapy and cell and gene therapy. So and this gene therapy is the application of biotechnology. This slide is showing the procedure by which the antibody and antigen, that is the interaction is showing that in case of ELISA or for diagnosis of any disease at earlier stage, the methods, that is ELISA, PCR, etc., which are the gift from biotechnology, are used. Here in diagram we can see the diagnosis for HIV, it may use these any of these methods, that is ELISA test or Western blots test or PCR test. Here the procedures are: antibody specific for a protein of interest is attached to a polymeric support such as a sheet of PVC. Then formation of antibody antigen complex, that means the interaction occurs between the antibody and antigen, and formation of second antibody antigen complex, and the amount of protein in sample is to amount of second antibody bound with the antigen. So this shows the diagnosis, this helps in diagnosis of particular disease.